Next, we are going to see about the organization of a chromosome hypothesis. This hypothesis is very important. There are several hypotheses to be made: a single-stranded, multi-stranded, folded. We have divided into two: a single-stranded, multi-stranded hypothesis and folded fiber hypothesis. But each are the individual hypothesis. This is first made hypothesis. This is second made hypothesis, fold fiber hypothesis, and nucleosome concept hypothesis. But among this, the nucleosome concept hypothesis is going to be accepted form of hypothesis. With the evidence, it was proved. So, this is the accepted form of hypothesis. As we said that about 5 to 10 μm diameter of nucleus is there, but there is a DNA which is going to be about 2 meter. Each DNA is about 2 meter. It is going to be compacted into this nucleus into the chromosome. This compaction, how this compaction is going to be taken place, is going to be detailed of and proposed by different hypotheses. These compacted chromosomes can be seen in the electron microscope as a fibril of 2 nm to 4 nm. When it is 2 nm to 4 nm chromosome, it can fit into the 5 to 10 μm of nucleus. A 2 meter DNA cannot be fit, but this can fit off. So the two hypotheses is going to be as single-stranded, multi-stranded or similar hypothesis, and folded fiber nucleosome are found to be similar. So I have made this proposed hypothesis into twice. But a nucleosome concept is going to be accepted model. A single-stranded, multi-stranded hypothesis. Single-stranded hypothesis, they propose that the DNA is going to be single-stranded. It is going to be converted into multi-stranded by lateral multiplication and tandem duplication. Lateral multiplication means there is a DNA. This DNA is going to be compacted by lateral, like a DNA. A next DNA is going to be attached laterally and increases multi-strandedness, which means the lateral addition of DNA is going to be taken place, and this causes us to make a chromosome. This is the proposed model of lateral multiplication. But in the tandem duplication, here we can say that a tandem duplication means also there is a multiplication of a DNA, where the DNA is going to be added off by increasing its length. Here the length is going to be increased off, but in the lateral multiplication, only the lateral multiplication is going to increase its sides. But in the tandem duplication, there is a length is going to be increased. These two types of proposed models in multi-stranded hypothesis. That means a multiple strand of DNA is going to be made, and this makes into a chromosome. This multiple strandedness is made by lateral multiplication or by tandem duplication. But this multiple strandedness hypothesis is going to lack the evidence, because even in the plant there is a single stranded nature of chromatin is going to be confirmed by the pulse field electrophoresis, which shows that there is no multi-strandedness. There is no multi-strandedness, which means there is no lateral multiplication or tandem duplication, because in the pulse field electrophoresis it shows that chromatin fibers are going to be single-stranded in nature. There is no multi-strandedness to be taken place. Therefore, this hypothesis is not accepted. The next proposed model is going to be folded fiber model, which was proposed by E.G. Dubray. He told that there is a histones there is a DNA, but a DNA is going to coiled or folded off. He didn't propose that how it going to be coiled, but he told that the DNA is going to be coiled into the chromosome, but coiling. And he also told that this coiling is made into the homogeneous diameter of 200 to 300 angstrom. But there is a histones. These histones are going to be outside in DNA, which makes it to make it coiling to be stable. As he said, there was a DNA, there was a coiling, but and also the histones make it stable. But here he missed the point that DNA is converted into a chromosome by coiling. So these histones are found to be outside the chromosome, which is going to stop it from functionality. But according to the accepted concept, the histones, some of them are found to be inside and some of them are found to be outside, which makes it to be stable chromosome according to nucleosome concept. So the folded fiber model is not accepted because the histone functions are not going to be described in correct manner, and also they told that there is a coiling to be occurs, which they does not explain that which makes it to be coiled. So the reason for the coiling is not explained in this model. So the folded fiber model by Dubray, proposed by Dubray, is not accepted. Then the nucleosome concept is proposed, which is it is going to be as accepted model of organization of the chromosome, organization of the gene. So the nucleosome concept is accepted. It is proposed by Audit and Kornberg. They proposed the nucleosome concept. It is accepted because they explained that histones are found to be octamer in inside, which makes them to start the organization of a DNA. Then the histones is also found in outside, which is going to be H1 in outside as a linker, and also a non-histones are also present, which is going to make it further organization. These are all explained by nucleosome concept. So the nucleosome concept is an accepted model. They also told that a DNA is going to form a histone helix on the supercoiled condition. It is very accepted model because of they explained that there is a supercoiled condition is going to be occurs. There is not a simple coiling to be occurs. There is a supercoiling occurs. Therefore, this is an accepted model. So next we are going to see about the levels of organization of the gene.